The following documentary is brought to you by 3 Briggs students who only wish to find the truth.
Fall of 2016, 3 students at Michigan State University set out to study the fight-or-flight response in both squirrels and humans, based on an experiment in the current zoology journal. We started by observing squirrel alertness based on different behaviors in 2 different locations.
One of the observation sites was located on the portion of the Red Cedar River which lies between Shaw Haw and Wills Haw. This is a high-stress location where the squirrels are surrounded by loud noises, buildings, and a high human density. We will classify human density as that there are at least 3 to 5 people within a 30-foot radius of the squirrel being observed.
The other observation site for this experiment was located behind Holmes Hall, both near the bird feeder that is strictly dedicated to observing and cannot be approached by humans, and in the Sanford Natural Area, the forest behind Holmes Hall. This was a very low-stress environment for squirrels because there are little to no threat that will alter squirrel behavior from visible or auditory distractions.
Experiments have been done to try to help understand what animal behaviors mean along with what behaviors coincide with each other. Our experiment was aimed at trying to depict if urban squirrels use behaviors that display more defensive and alert manners because they view their surroundings as potential threats, while squirrels in a rural environment display behaviors linked to being relaxed. We did this by conducting an observational experiment. To conduct this study on behaviors displayed by squirrels in different locations, we decided to focus on squirrel vigilance and squirrel tail movement.
When looking at stance, there is either bipedal or quadrupedal. Bipedal is when the squirrel is observed standing on only its 2 back feet. Quadrupedal is when the squirrel is seen on all 4s. The tail is also observed during this experiment. We will discern whether squirrels are using a tail flicking or a tail flagging movement. Tail flicking is shown when the tail moves fast at medium power for a short period of time and only the end portion of the tail is moving. Tail flagging is seen when the tail waves at high power, usually the tail moves above their heads.
These are some examples of tail flicking. Note how the tail does not surpass the head and how quick it is done. It is believed that flicking is demonstrated by squirrels that are more relaxed in their environment.
Tail flagging is shown when squirrels are in a more defensive mode. They use flagging as a way to communicate to other squirrels about potential nearby threats. This movement is done multiple times at a very high power, mostly while in a bipedal stance. We believe from our observations that tail flagging and a bipedal stance coincide as vigilant behavior.
This is the bipedal stance believed to be associated with defensive behavior.
The quadrupedal stance is shown when the squirrel is more relaxed, generally associated with tail flicking.
Humans and squirrels both possess the PNMT gene. It is a protein-coding gene found on chromosome 17. This gene is activated by stress hormones. The protein PNMT methylates the amine in norepinephrine, creating epinephrine. The PNMT gene plays a key role in regulating the epinephrine production. Epinephrine is adrenaline. Adrenaline is a hormone secreted into the bloodstream by the adrenal gland, especially in conditions of high stress and fear.
In our human experiment, behaviors of fight, flight, flinching, and freezing were observed. To test for this, we observed human subjects in both heavily populated situations and one alone.
Through the many observations and data that was collected, we found that squirrels in urban environments showed both alert and inattentive behaviors, depending on the proximity and interaction of the threat, while squirrels in a rural environment showed only inattentive behaviors.
For our human experiment, we found that the subjects that were tested in a solitary environment were more likely to react in the form of flinching or freezing than those that were in areas that were highly populated. This evidence supported our hypothesis.
When comparing the human and squirrel data, we saw that there was a corresponding trend to the movements that were made when in high-stress environments and when in low-stress environments. We can relate this behavior back to the PNMT gene that is present in both humans and squirrels because it is responsible for releasing adrenaline into the bloodstream during fight or flight situations.
Hopefully, further research of behaviors of both squirrels and humans will lead to many years of peace and harmony on Michigan State campus.